This introductory slide shows the Genetics Institute building at the University of Florida. The water in the foreground is Lake Alice, which is a wildlife refuge, and the lake is full of alligators, which sometimes wander around the campus, and so we're all very alert at all times. Okay. Gene therapy, the notion of gene therapy really got started a little over 20 years ago. Molecular genetics had probably been the most exciting area of biology since Watson and Crick for about 40 years. And as things progressed, the thought was that we ought to be able to actually apply some of this knowledge that had been gained to treat people. And so the notion of gene therapy was advanced. There was a lot of hype, and yet 20 years after we started working on gene therapy, we're just getting to the point where we think it actually works. And I think that there are several lessons, and it's the lessons that I'd sort of like to go over this morning. So we need three things in order to do gene therapy. The first thing is that the genetic lesion has to be defined. We have to know what's wrong. Then we need a supply of the wild-type gene. Those two items are no problem. We have lots of information there. And then we need some way to deliver the gene to the patient in a manner which allows it to function. And it's this last step which has proven to be a really major challenge for the field. So if we think about what's required in order for successful gene transfer to occur, a good vector which carries the transgene has to be able to get to the right cell. It has to be able to attach, to get into the cell, and then be successfully translocated into the nucleus, and then appropriate expression has to occur. And in addition to having to achieve those steps, there are two other concerns that we have. One is whether or not the vector itself is toxic, or whether the expression of the transgene might be toxic. And it's an interesting theoretical consideration because many of the genetic diseases result from null mutations, and so there's no expression of the protein. And the question then is, if a new protein is expressed, is it going to be recognized as a foreign protein and rejected by the body of the patient? Interestingly enough, that has not turned out to be a major problem so far, but it's something that we always have to consider. Another issue which has been raised is whether or not too much of the transgene might be expressed. Can we really regulate it in a way that's comparable to the physiological process? Interestingly enough, that also has not turned out to be too much of a challenge. More of a challenge has been if the transgene starts to be expressed and the product made, does it continue being made? And in the case of most vectors, it turns out that expression has been fairly short-lived. And one of the really nice things about AAV vectors is that they keep on expressing. And I'll give you a couple of examples in a little bit where that expression has extended for quite a few years. So there are two approaches to gene therapy. You can either inject the gene directly into the patient, or you can remove the target cells from the patient, grow them in cell culture, modify them, and put them back into the patient. And both approaches have been used. The French have been very successful in ex vivo gene therapy for children with severe combined immunodeficiency disease. And the examples I'm going to show you today take the other approach of injecting the vector directly into the patient. The other consideration is whether or not the vector exists as an extra chromosomal element or gets integrated into the host cell DNA. And the point is that if the cells that are the target cells are post-mitotic and they're not turning over rapidly, then extra chromosomal maintenance of the vector is perfectly reasonable and in fact has worked in several instances. On the other hand, if there's reasonable turnover of the tissue, then an extra chromosomal vector probably is not going to be useful in the long run. So the question is, what makes a good vector? All sorts of approaches to vectors have been tried, but what's become clear is that although all of those approaches work at some level in the test tube, in terms of actually being practical, being efficient enough to make a difference, the one vector that probably is useful so far has been viral vectors. And because if you think about viruses, they are vectors. They introduce nucleic acids into cells, and those nucleic acids are expressed. The problem, of course, is that they cause diseases for the most part. And so somehow you have to modify them so they'll express what you're interested in and not cause a problem for the patient. So of all of the viruses that are listed here, Adeno-associated virus, or AAV, has been one of the more successful ones, and I'm going to talk about that today. On coronaviruses, or murine retroviruses, have been used, again, in the case of the French experiments on severe combined immunodeficiency disease, but they illustrate the problem that we all worry about, and that is they were successful in that experiment. Patients who otherwise would have died were essentially cured. 12 were treated in the first go-round. Unfortunately, 4 of them developed leukemia because that's what this viral vector tends to do naturally. We've gotten somewhat better about avoiding that, but the interesting thing to me was the reaction of the French government. I think in the States we would have just said you can't do this kind of thing, you can't give kids leukemia. But the French said, you know, here are 12 kids who are going to die. It's better that 8 of them do okay and 4 of them get leukemia. And of course, things were improved because we can treat leukemia. And so 3 of the 4 at least had their leukemia remitted successfully. And the experiments continued. I think one of the great triumphs, actually, of gene therapy. Lentiviruses have advantages, except for the fact that they come from HIV, and there's some always political questions about using that kind of vector, and the same thing is true of poxvirus vectors. So I'm going to talk about adeno-associated virus, and I think a lot of talks this morning are going to involve AAV, so it's not exactly a mystery to everybody. It's a very small virus. It won't replicate in culture unless there's a helper virus. But just for fun, can you raise your hand if you've been infected by AAV naturally? See, a couple of people know what's going on here, that's clear. Well, it turns out that about 90% of us are seropositive, which illustrates why this is such an interesting vector. It infects people routinely doesn't cause any disease. So we start off with an enormous advantage biologically speaking. Now, there are numerous serotypes. There's at least 10 that have been identified and described for people, and in non-human primates, there are over 100 which have been detected. All of these are potentially vectors for gene therapy. Okay, so the life cycle, in very simple terms, is illustrated in this slide. If there's a helper virus co-infection, productive replication goes on and progeny virus are made. If there is no helper virus, then the DNA of AAV gets integrated into a specific site on chromosome 19 in dividing cells, and it's maintained there in a latent form until the cell is stressed, usually by superinfection with a helper virus. And then the integrated genome gets activated and AAV is produced. And how long can this go on? In cell culture, it's possible to pass cells more than 100 times and still maintain the integrated state and be able to rescue the latent genome. So it's quite a stable situation, although there is some dynamic aspect to it, and every now and then a genome will pop out just under normal circumstances. So recombinant AAV vectors are kind of interesting. One of the issues that's kind of nice is that AAV vectors essentially only contain the terminal 200 nucleotides of the AAV genome, and everything else in between the terminal repeats is what you put into the vector. And so that usually includes the transgene and any regulatory elements that are going to be put in. And producing vector conceptually is simple. You have to put in the transgene on a plasmid between two copies of the AAV terminal repeat. You have to put in the helper genes from adenovirus or herpes virus, and you have to put in the normal AAV genes, which are required for replication. But the plasmid that's labeled AAV contains none of the terminal repeat sequences. So the terminal repeat sequences are required for packaging the viral DNA, and the way this is set up is that there's no homology among these three plasmids. So what gets packaged and comes out as viral vectors should only be the vector itself. There should be no wild-type virus. And much of the time, that's what happens. But this virus is very wily. And so in spite of the fact that you designed the situation so that there's no possible recombination, so that wild-type virus could ensue, in some cases it happens anyhow. And so one of the critical quality control measures on every batch that's made for clinical use is to make sure that you can't detect wild-type AAV. Now, the interesting thing, of course, is that in truth, it probably really wouldn't make any difference at all, but from the point of view of our Food and Drug Administration, they would like pure preparations administered to the patients. Okay, so what I want to talk to you about today are several issues. One is called Leber's congenital amaurosis. That's a congenital form of blindness. Hemophilia B, and then structural modification of the vectors, which have been done recently, and then where we may be going in the future. So the first thing that I want to talk about is Leber's congenital amaurosis. And I'll just say that again that this is a form of childhood blindness. Kids who have it go blind usually in the first year or two of life, and they're essentially really blind. And what we do in this case is to introduce a vector which contains the wild type form of the defective protein, which is called RPE65, subretinally. And this is the actual biochemistry of the situation. RPE65 takes place in the retinal pigment epithelium cells, and it's involved in the conversion of all trans-retinal ester to 11-cis-retinol. And then, oops, God, what have I done here? Okay. And that blocks then essentially the rhodopsin cycle, which involves not only the retinal pigment epithelium, but the photoreceptor cells, primarily the rods. And so essentially light cannot be converted into a signal in the central nervous system. Okay. So the question was, if you simply put in a vector, as I've described here, what would happen? And in animal models, it turned out that this experiment was very successful. There's a strain of dogs, the Briard, large dog, which have this disease. And it turns out that if you treat these dogs shortly after birth in one eye, they can see for the length of their expected life. And the first dog was named Lancelot, and he survived for more than 10 years, and one shot essentially gave him vision in his treated eye for that period of time. Lancelot was very popular. He was taken to the US Senate, where he testified on behalf of the Eye Institute on several occasions, successfully, I might say. So that was good. But of course, the trouble is, we all know that animal models are necessary and they have to work, but it doesn't tell you whether or not it's going to work for people. And so it came to the clinical trial, and one I'm describing here was done by people from the University of Florida and the University of Pennsylvania. And it's been going now for 3 years, and 15 people were treated to make sure that there was no toxicity. And what they found was that of the 15 people who were treated, all of them gained an increase in full field light sensitivity. And if they just looked at the area that was actually treated of the retina, that light sensitivity had increased anywhere between 200 and 60,000 fold. More interestingly, most of them regained a pupillary light reflex, and 5 of 6 who were tested actually could make their way through a maze without bumping into things too much. And their visual acuity increased. And I think the interesting thing, there were 2 in nature. One is that there was no toxicity whatsoever detected, and the therapy was long-lasting. Okay. So, and the patients actually are happy in most cases. One guy complained because the light on his clock radio was too bright, but one saw fireflies for the first time. One claimed that he had forgotten how blue the sky is, and et cetera. So it was successful. Now, I think one of the interesting things, which isn't on the slide, is that these patients were not treated in the area of the fovea. So you wouldn't expect any improvement in color vision. And yet a fair number of them actually regained color vision. And the treatment area was close, but I think what it illustrates is the plasticity of the brain and the fact that we really don't understand all the details that are involved at that level. But there's no question that these people regained a certain amount of color vision. The second problem I want to talk to you about briefly is hemophilia B, which is due to a defect in factor IX. That gene is small enough to insert into AAV vectors. And in animal models, it worked very well, and right up to the level of the dog. Dogs with hemophilia B were cured. And then they did a phase 1 trial at Penn, and there was a total failure. Okay, there was very low level of expression of factor IX, and not only was there a low level of expression, but in the only people who really expressed detectable levels in the serum, which were short-lived, they in fact had evidence of an inflammatory reaction in the liver. So that was very disconcerting. A lot of effort went into figuring out what was going on. And the best guess is that enough vector had to be put in so that the vector itself, which remained on the surface of the hepatocytes, or the products of the breakdown of the vector, which were expressed in the MHC system, induced the inflammatory response. So a better vector was clearly required if there was any hope. And I'll just skip over that one. And recently, and just before Christmas last year, there was a paper that came out of London and also with the Penn group saying that they had actually now been more successful in curing hemophilia B, and they used a different AAV serotype, AAV8, which is much more effective at transducing hepatocytes. And actually, they simply injected this vector in peripheral vessels and still got a good result. Now, the question is that the full answer? And unfortunately, like most of this, it's not. And the problem is that if one looks at what kind of immune response they got at higher doses where there was a better result, they started seeing inflammatory reactions once again. And so it's clear that there's still room for improvement. And what I want to talk to you about very briefly is the fact that work has been done by Arun Srivastava at Florida. And what he's found is that when the AAV vector gets into the cell and gets released from the cytoplasmic vesicles, that ubiquitin tends to get hooked onto the surface tyrosines. And when that happens sufficiently, the vector goes to the proteasome and is degraded rather than going to the nucleus and being expressed. And so what he figured out, with the help of Mavis Agbanji McKenna, who's a great crystallographer, was which tyrosines were on the cell surface. So he simply substituted phenylalanine for tyrosine. And when he did that, it turned out that the efficiency of the vector went up enormously. And this paper simply shows that in a mouse model, they got very good transduction. So the summary is that you can do site-directed mutagenesis to get rid of the tyrosines. You can increase the efficiency by 30-fold, and you get therapeutic levels of human factor IX in the animal model. And so the goal at this point is to go into the dogs and then go back into people. Okay. So far, I've sort of given you a decreasing list of success stories, but like every other series of clinical trials, there was a disaster. And this was a trial with rheumatoid arthritis, where the notion was that if you could put an AAV vector, which would block TNF receptors into a hot joint, that you could really locally alleviate the effects of rheumatoid arthritis. That was tried. The first patient that tried in was a middle-aged woman who about 2 days after the injection got sick and several days later died. So that was a real bad outcome. And they tried to figure out what had happened. And it turned out that what had happened was that this woman had died of overwhelming histoplasmosis. And when they tried to figure out how this had happened, it turned out that when they went back to old serum samples before the trial started, that in fact she had a low-level infection. She'd been treated with systemic TNF inhibitors, which were then stopped before the trial. And the conclusion was, unfortunately, or fortunately for AAV, but not for the woman, that the problem had nothing to do with the trial per se, but was a consequence of the systemic anti-TNF receptor therapy. But I will tell you that trial has not been repeated subsequently. So, where are we going with future AAV vectors? I'll go by the nanoparticle mantra that everybody uses. The notion is, can you make targeted AAV vectors both for the cells, and people are working on that. And a different kind of targeting is that you can flank the transgene with ribosomal DNA sequences. There are lots of copies of the ribosomal genes in the cell, and you really tend to cause an increased level of integration, it seems to be, into the ribosomal genes, and that turns out to be a fairly benign site of integration. And so you can try to then use AAV as a vector in dividing cells. I mean, currently, from the way what I described to you, all AAV vectors are non-integrating because the rep gene, which encodes the regulatory protein of AAV, is missing from the vectors, and the rep gene is required for site-specific integration. It's also possible to design multi-component vector systems where rep is on a separate genome from the transgene, so you can get site-specific integration under those conditions. And my guess is that in the future we'll be working quite a lot in these areas. So I would just like to end by giving you a couple take-home messages. The first is that 40 some odd years ago, my lab started working with AAV. This was pure basic research. No idea of curing blindness, okay? And I think that there's a message here, both for funding agencies and scientists and clinicians, and that is that you never know with basic research where it's going to lead. Almost all of the good things that have happened have come about, obviously, as a consequence of this kind of research. Related to that, in fact, is the history of gene therapy. People, once they got the idea of gene therapy, raced into the clinic to try it in clinical trials, all of which didn't seem to work very well. And what the message that has become clear as we go through this and begin to have some success is that if you're really going to be successful at doing gene therapy, you really have to understand the fundamental biology of the virus and the way it interacts in the human body. So on that note, I'll thank you very much for your attention.